This is the second part of olfaction, so our sense of smell, and we are continuing on through that chapter called the chemical senses. So I usually start out with moving to the next slide where there's a picture and then coming back and making sure I said what I wanted to say. And I have this horrible tendency when I'm looking at these slides to see what's on the slide and get very distracted actually by the words, as I'm usually not looking at the words anymore as I'm talking to my class. But here we are. And some of this is going to be repeated because I usually do this with the picture, and I'm going to continue to say what I want to say with the picture. But our olfactory mucosa are located in the upper region of the nasal cavity. They are made up of the olfactory epithelium, if that sounds like pigment epithelium. That is the back of the retina in the eye. The olfactory epithelium is basically like the retina of the nose, and that includes the retina of the nose includes the underlying tissue, the blood vessels, the glands, and the bundles of axon fibers from the olfactory receptor neurons. Okay, and it protects the olfactory mucosa, protects the olfactory epithelium and allows odors to dissolve. So it's kind of moist up there, and it allows the odors to dissolve so that they can be detected by the olfactory receptor neurons. The olfactory receptor neurons are a lot like the rods and cones of the retina, and they are similar to the rods and cones in a number of ways. First, they contain those molecules that are sensitive to odorants, and this is similar to how the rods and cones have the visual pigment molecules that are sensitive to electromagnetic stimulation. They are made up of proteins that cross the membrane of the receptor neuron 7 times. And like rods and cones, they are sensitive to a range of specific stimuli. We're going to get back to that idea of the puzzle of olfactory quality, because while we have rods and 3 types of cones, and they are each responding, these 4 types of sensory receptors are responding to some range of the electromagnetic radiation that is close to us in the same area of electromagnetic radiation. We have 350 different types of olfactory receptor neurons, and for us to tell what a smell is, we're going to look at the pattern across 350 types of these different kinds of receptors. And just for comparison, there are 1,000 different types in mice, so they can detect, if we can detect, according to this one piece of research, supposedly about 1 trillion different odors, they can detect that many more because they have that many more different types of receptors, just like we saw with the animals that have 4 or 5 or 6 different cones, or I can't remember how many these particular kinds of shrimp have, something like 100 different cones. They're going to see a lot more variation in colors. These mice are going to smell a lot more variations in odorants. And I do have my word art up there to say this is where transduction is taking place, right, in these molecules that are sensitive to odorants. So transduction, I'm singing it so that we remember it's this wonderful magical thing that allows us to experience the environmental world. So here's my picture, and I'm going to repeat a lot of what I just said with a couple more pieces of information, and then I think quickly go through some of the future information that is repeating this, but probably more quickly because I do like to talk from a picture. But this is showing the olfactory mucosa, that area up there that is made up of a bunch of different cells. It has the cilia, or those areas of the olfactory receptors that are sticking out, that are going to actually be activated by odorants. It is moist up in there, and there are other cells that are giving us the support structure. Okay, those olfactory receptor neurons are going to communicate through the olfactory nerve. We have seen some other nerves here, right? The optic nerve and the auditory nerve. The olfactory nerve is going to communicate to the olfactory bulb, which you can see lies just above this little cribiform plate. So that kind of hard plating in your nose, your olfactory bulb is up and behind that. And those olfactory receptor neurons, what Goldstein, I think, does a really neat job of showing with this picture is that If you look at the green versus the purple versus the orange, those are giving you examples of, let's say we have 3 different kinds of types of olfactory receptor neurons. Remember, there are 350. So we're keeping this really simple with 3. And those are going to communicate up through the olfactory nerve. And let's just say, so if I'm a purple olfactory receptor neuron, I am a particular type of olfactory receptor neuron, and I'm going to communicate to what appears to be this little purple gem up there. Well, that little purple gem up in the olfactory bulb, that is actually a glomerulus. And so if I'm a purple olfactory receptor neuron, I'm going to communicate to probably a pair of glomeruli. So 2 of these little glomeruluses, or glomeruli, are going to then be activated in the olfactory bulb. And so we have a particular type of olfactory receptor neuron communicating to a particular pair of glomeruli in the olfactory bulb. So we're now looking at recognition profiles of various odorants, and what this is showing us is the particular olfactory receptor neurons that are active and just how active they are. So a large circle is that they are highly active, and the small circle there is that it is slightly active. And if you'll notice over to the right, our molecules, they look relatively similar, but remember the puzzle of olfactory quality is that even though we have similar molecules, they might have really different perceptions. We might have molecules that are very different from each other, and we're going to might have a similar perception. And what that perception, what that experience of the smell depends on is this recognition profile. And so he compares octanoic acid to octanol. Octanol is often described as a rose, kind of fresh, sweet smell, and octanoic acid is described often as rancid, or oh, I always forget what he calls it, sour, repulsive. And if we look at the recognition profiles for octanoic acid, we have olfactory receptor neurons 1, 18, 19, 41, 46, 51, and 83 are highly active, and then 79 also has some little bit of activity. And octanol looks really different, so 18, 19, 41, and 51. And this is just an example with a few of the olfactory receptor neurons, but you'd see that these would cause a different pattern of activation at the level of the olfactory receptor neurons. And here's just the language that of what I just said, but in words. Those molecules with similar structure can smell different. They can have different recognition profiles. The example of octanol and octanoic acid, and that they have different recognition profiles. This is what's causing this really difference in perception. So just as a fun comparative, I got this information from Dr. Neil Levins. The bloodhound has about 220 million olfactory receptor neurons and 5% of cortex devoted to olfaction, compared to humans who have about 20 million olfactory receptor neurons and about 0.1% of the cortex devoted to olfaction. If you remember, so we talked about this for particular sense in the sense of a particular sensory modality, but if I have cortical magnification of something like my fingers, it gives me more acuity, more ability to feel detail. And I see the same thing with my vision, right? As we have cortical magnification of the fovea, and that's where I have the most detailed vision. So if we notice that they have 5% of their cortex compared to 0.1% for us, they have a lot more acuity or ability to smell. And while we can detect about the same number of smells as dogs, they can detect concentrations about 100 million times lower. And all of these, you might notice that my estimate here of humans my estimate before of humans was about 10 million olfactory receptor neurons, and I think we said something like 1 billion for olfactory receptor neurons for dogs. If that's not in a previous slide, it's in the book. And just to notice that these are estimates from different people doing research. If we go back to our picture, I've already said some of this, as the axons of those olfactory receptor neurons are going to pass through that bony structure called the cribriform plate and bundle to form the olfactory nerve. They're going through that cribriform plate and that olfactory nerve is, and then it's entering the olfactory bulb. And again, much of this is repeating from the previous picture, but here I have another picture that shows nicely kind of what's happening with the glomeruli on the olfactory bulb, where we see this convergence of about 20 million olfactory receptor neurons are going to converge onto 6,000 glomeruli. And we've talked about what convergence gives us before when we talked about the rods and how they communicate. And as I mentioned before, with the green and the purple and the orange, but now he gives us blue and red, those olfactory receptor neurons that are expressing a specific type, olfactory receptor neuron type, they're going to converge onto one glomerulus pair. Okay, so a pair of glomeruli. So if we look at just as an example, if we look at the olfactory receptor neurons that respond to methyl salicylate, those are going to send axons to a single glomerulus pair. So we've been talking about what's happening at the level of the olfactory receptor neurons, and that we have this recognition profile and certain types of neurons responding to certain chemicals, and that what's really going to be changing in that recognition profile is going to those different recognition profiles. If they're very different, it's going to mean different perception. And what we're seeing at the olfactory bulb, it's going to show activation of different areas for different chemicals, and as we have the molecules, the chemicals, the chain length increasing, we see that area on this picture as moving farther to the left. And so if you see from the dark red at the very top that that's a very short chain length, and as we move down to the blues, getting a longer and longer chain length, that's that activation as measured by oxygen consumption. So where there's less oxygen, we see that moving to the left on the olfactory bulb. But remember that part of what's important is the perception. And so what's happening, what we're going to see is the pattern of activation on the olfactory bulb. It's going to reflect the different chemicals and the chain lengths, as we just saw, but it's also going to reflect different perceptions. And so I'm going to start with the ones that are the same. So if we move over to the right, the plus or minus limonene, those are very similar in perception. They're very similar in their chemicals and chain length, and we see very similar patterns of activation. If we look at the plus or minus carvone, those are very similar in their chemicals and their chain length, but now, or what the molecules look like and the chemicals look like in the chain length, but now we have different perceptions, and we can see this as there is a great deal of similarity across the olfactory bulb, but we see specific differences as well. And so what he's pointing to with the white arrows there is the one on the left in A. We have more activation up at the top and then over to the mid-right, and the one to the right in A that has a different perception. We have less activation of what we just saw, of where those arrows were pointing, and now this white arrow is pointing of where we have a lot of activation in this example that we did not have in the one on the left. And so we're seeing similarity because of the chemicals and the chain length, but there are differences because we also need to code for the difference in perception. And so at the olfactory bulb, we're going to call this a chemotopic map, an odor map, sometimes called an ototopic map. But I think I'm pretty good at just sticking to chemotopic map, as it's a map. It's a chemically based map in this case. If you remember, we've had our primary receiving areas having something like a retinotopic map or a tonotopic map. And for sometimes we saw that as going through those subcortical structures. Here we are in the olfactory bulb, the very first place where olfaction synapses after being experienced by the sensory receptors. Okay, and as we look at what's happening here, the olfactory mucosa, so where the olfactory receptor neurons, those sensory neurons are communicating to the olfactory bulb, and the olfactory bulb is communicating directly to the piriform cortex, which is the primary receiving area for olfaction. So the primary olfactory cortex is called the piriform cortex. But look, it's not just communicating directly with the piriform cortex, so i.e. not going through the thalamus like we have seen for every other sensory modality so far. It's also communicating directly with the amygdala. And actually, so usually we read a journal article here about the sense of smell and about the piriform cortex and its mapping, that it is communicating across the entire hemisphere to all of those limbic system regions, the entorhinal cortex, the hippocampus, the amygdala, and the orbitofrontal cortex. And it has these neurons that, instead of communicating to other neurons close by that are doing something similar, they are communicating across the hemisphere to thousands of neurons. And the neuron next to me, if I'm a particular neuron, is not going to necessarily be communicating to the same neurons that I'm communicating to, or even neurons that are close to what the neurons that I'm communicating to. In fact, they looked at two neurons close to each other in the piriform cortex, and they were communicating to, they had almost no overlap in the neurons that they were communicating to. And again, thousands of neurons across the hemisphere. And so what's happening, as we're going to talk about this experience of olfaction, and I've already commented on how we don't remember the names of what we're smelling very well until somebody tells us the name, and then the quality actually can change based on that experience. But what we often remember with the sense of smell is memories, and especially emotionally associated memories. And it has this vivid sense where we tend to feel some sense of emotion when we are smelling something. And that is because of this. It's not going through the thalamus. It's not going to these higher level visual areas that are then, or visual or other sensory areas that are doing the same kind of information processing. It is going throughout the hemisphere directly from the olfactory bulb, which comes directly from the olfactory receptors themselves. There is evidence that the link between a stimulus or a particular odor and behavior is hardwired. We at least see this in other animals, but I'm sure there are hardwired links for humans as well, just maybe more difficult to look at. But so rats, they have exposed a rat to a cat odor, and the rat has a triggered fear response. And that's true even if those rats are brought up in a lab and have never seen a cat. And that's true even if those rats have been brought up in a lab for a number of generations, where the generations have never seen a cat. So this is not something that's learned. This is something that is hardwired and innate in rats to have this triggered fear response. Another example of pheromone responses, so there's a chemical released by female rabbits that triggers nursing behavior in newborn rabbits. And a lot of times we talk about nursing behavior as being as there's a lot of classical conditioning there, but there's also some innate hardwired responses. And higher order responses are going to occur later in processing in the cortex. And we're going to see that these more complicated responses are not so hardwired. And so we are very much influenced by our higher cortical processes. And all of this processing happens so quickly that, again, we're not aware of being hardwired in any way. And yet I'm sure we are, but we also have very quickly this higher order processing that is giving us more complicated responses that are not hardwired.